So, this is part 4 of our lecture on Chapter 43 on the immune system. And as you can tell, there's a lot to cover, and the immune system is really complicated. And so, we're going to be going over the level that you really need to be familiar with. And I'm sure there will be lots of questions that come up and that you can pursue with future courses that will go into so much more detail. But for now, I'm trying to keep this at a level that will be helpful for you for this course. So, how antibodies work, they do one of a number of things. They can neutralize a pathogen by binding it. So, in this first box here, you can see how they come around and they surround the pathogen. And so the antibodies essentially make it inactive in this way. The other thing that they do is they can come together in agglutinate particles. So these little oblong structures are bacteria that you see here. And in the middle, we have a number of antibodies working together. And what they're doing basically is clumping the bacteria together, which then makes it easier for a macrophage to come along and engulf this structure and at the same time take out a whole group of bacteria. They can also cause the precipitation of a soluble antigen by binding to it and making it become more soluble. And again, each of these three activities are going to help the macrophage eat up the tagged invaders, essentially. The last way that an antibody works is it can activate the complement proteins. So, for example, you see here, this is a foreign cell, and what's attached to it is an antibody here that is then sending out a signal to complement proteins to come and cause the lysis of this foreign cell. There are general classes of antibodies. So again, we can have millions of different antigens, and so therefore we need millions of different types of antibodies. But we group them into classes. And they, again, another word for an antibody is an immunoglobulin. So the classes are called the IgGs, the IgM, IgG, IgA, IgE, and IgD. So these different classes actually have some really general functions that are how they're classified. So the two primary types that we find circulating in the blood are IgM and IgD. So IgM is responsible for the first immune response, and they activate complement proteins. So those are the main jobs of these types. They can then go on to actually what we call isotope switch. And so they can actually go on to produce and be transformed into these other types of antibodies. So we can have an IgM become an IgG, which is a second response antibody. So this is, again, a major type that circulates in the plasma, and its main job is to promote phagocytosis. IgAs you would find in external secretions, and IgEs promote the release of histamines and bodily fluids. They actually came about as a reaction to parasites, and these are the ones that many of us are familiar with because these are the ones that trigger allergic reactions. So how does the B cell respond to an infection? It actually takes about 10 to 17 days for a full response to an infection. So it's not a quick process. So this is a first-time infection by a foreign antigen or invader. And so what's going to happen is the B cells are going to notice this invader. And so what they're going to do is then start to, so what will happen is this invader will be a foreign antigen. The B cells will have receptors on their surface which actually bind to the antigens that are on this invader. The binding on the B cell then activates that B cell. So that B cell is called an effector cell, and they will start to go through a lot of cell division. So we have these B cells with antibodies, we have the binding here. It'll start to make thousands of clones of this particular cell. And this is again the B cell with the correct receptors on it. And so these receptors are then activated. We have the multiplication of these cells occurring. And then these effector forms of the B cell are actually what we call plasma cells, because these are the ones that are going to release antibodies. Now, so these plasma cells are releasing the antibodies and producing them into the system to now attack in one of those four different ways we saw earlier. In this case, they're binding on the outside to tag it for the macrophages. And then also a portion of these cells are going to become memory cells. And so memory cells are long-lived cells that produce an effector cell like this, these plasma cells. Effector with an E means that they're actually active cells. These memory cells can be, whenever they sense the presence of this foreign antigen again, they will immediately go into making the plasma cells, or the plasma B cells. So we skip these other steps here. So we have a much quicker response. So how do vertebrates produce millions of antibody proteins if they only have a few hundred genes to code for those proteins? And the way that they do this is by rearranging the DNA. So we haven't really gone into our genetics unit yet, but in the processing, once DNA has been transcribed, there is a process where it is actually spliced into different forms. And so what happens with the genes that are coding for the different antibodies or B cell receptors in particular, that is all done through this rearrangement of DNA. So that's how we get our different types of differentiated B cells. Now, those memory B cells are what are responsible for the effectiveness of a vaccination. So when, again, an immune system is exposed to a harmless version of the pathogen or a harmful version, we get an active immunity or an acquired immunity response. We can also get that same response sometimes when there is a harmless version of the pathogen, or, for example, a partial portion of the pathogen that can't really cause infection, but it still is recognized by B cells in a way that triggers the production of memory B cells. So the immune system gets stimulated to produce antibodies, and then those memory B cells are floating around forever, waiting for any possible attack by this particular invader in the future. We've found that vaccinations are most effective against viruses. And this is just a picture of a very famous scientist, Jonas Salk, who developed the first vaccine. And the vaccine that he developed was against polio, which was a devastating illness that was common, very common actually, several decades ago. In this particular polio, 1 in 200 infections led to a permanent paralysis. And then among those, 5 to 10% died when their breathing muscles were immobilized. So this vaccine had a huge beneficial effect. And it was actually developed by using an inactivated form of the polio virus that was actually grown in monkey kidney tissue and then inactivated with a chemical and put into this vaccine, which has proven to be very effective in helping the immune system to never succumb to the polio virus. So what we've been talking about is active or acquired immunity. And actually, we can have an acquired immunity through a more passive route. So we can have active immunity where we actually had the infection, either through a vaccination of a live form of the, or an inactive form of the pathogen that causes our immune system to actually work to produce its own antibodies. But we can also have passive immunity in two different ways. We get passive immunity when we get antibodies from somebody else. So you can have a vaccine that actually injects you with antibodies that have been produced by another person or organism, and that would give you a short-term immunity. You can also, this type of immunity is what newborns have. So when a mother breastfeeds their child, then they are actually giving their child their own antibodies through the breast milk. Also, when a baby is in utero, antibodies from the mother are passed directly across the placenta into the baby. And so the placenta actually is providing a very short-term collection of antibodies for the baby to protect it in its first weeks of life. So now we've talked about B cells, but what about T cells or T lymphocytes? So again, the difference between these, the B cells were a humoral response. That means they were in the tissue, in the fluids floating around. T cells, on the other hand, are cell-mediated response. They only are responding and dealing with infected cells. And so cells that have viruses, bacteria, parasites that have entered them. So these are our somatic, our body cells that actually have the invaders within them. So now the T cells are going to react to that. So this is a defense against non-self cells. Also, so cancer and transplant cells are also going to be a target of a T cell. So T cells are targeting infected cells and non-self cells. There are two types of T cells. There are helper T cells, and there are killer or cytotoxic T cells. So how are cells tagged with antigens? So what a T cell is going to do is it's going to look for a cell that has been infected. And how does it know it's infected? It does that by noticing what are called antigen presenting portions on the outside of that infected cell. So the T cell has receptors on it just like a B cell, but their receptors are actually looking for what are called MHC, or major histocompatibility proteins. These proteins are on the outside of an infected cell, just on the membrane. And what they are the portions of the antigen that have been broken down within the cell are either placed onto these